morning children. Welcome back to online class today. Children, I hope you all completed the previous portion what I taught already. Okay children, in the previous class we all learned about the types of chromosomes based on the position of the centromere, based on the function and the structure of DNA. Isn't it now? That what are the types of chromosome based on the position of the centromere? Telocentric, acrocentric, metacentric and submetacentric. Isn't it mom? What are the types of chromosome based on their function? Allosomes and autosomes. Allosomes are also called sex chromosomes. The number of chromosomes present in your body is 46, isn't it? That means 23 pairs. Okay. So out of 23 pairs, 22 pairs determine our body character or somatic character. So they are called autosomes. Isn't it mom? Or somatic chromosomes. The 23rd pair which determine the sex of an individual. That is called allosomes or sex chromosomes. Isn't it mom? So types of chromosomes based on their function. There are two types of chromosomes based on their function, allosomes and autosomes. So in the previous class we all already learned about the structure of DNA also, isn't it mom? So who were proposed the double helical structure of DNA? James Watson and Francis Crick. DNA consists of millions of nucleotides. So we call them as polynucleotide chain, isn't it? It consists of nucleotides. What are called nucleotides? The nucleoside with the phosphate group is called nucleotide. What do you mean by nucleoside? Nitrogenous base combines with sugar molecule and form nucleoside, isn't it mom? So DNA consists of polynucleotides. What are the two nitrogenous bases found in DNA? Purines and pyrimidines. What are the purine bases? Adenine and guanine. What are the pyrimidine bases? Thymine and cytosine. Isn't it? So base pair in the DNA is specific. You all know that how it is. Adenine always pairs with thymine. Guanine always pairs with cytosine with triple bonds. The bond between adenine and thymine is double bond, isn't it mom? Today we are going to learn about DNA replication. Okay, DNA replication. This replication is a process that takes place within the cell. Okay, it is one of the basic process that occurs within the cell. In this process, the DNA molecule produces its DNA. It's the structure of this is the structure of DNA. This DNA molecule produces the exact copies of its own structure. So during replication, replication is one of the basic process. That means the DNA molecule produces its exact copies or its own copies during this process. Okay, it consists of two strands, polynucleotide strands. You know that. So this is one of the strands. This is another strand, isn't it, ma? The two strands of DNA molecule have a complementary base pairs. You know that complementary base pairs in the sense this base is present in DNA are specific. Adenine pairs with thymine and guanine pairs with cytosine, isn't it? So the two strands of DNA molecule have a complementary base pairs. The nucleotides of each strand provide the information needed to produce its new strand. So replication means the DNA produces own copies or the exact copies. So the nucleotides present in the DNA have the information or to provide the information needed to produce its new strand. Okay. So what about the daughter DNA? The daughter DNA contain the same genetic information as there in the parent, isn't it mom? So DNA produces own copies. So what about the daughters? The resulting daughter cells contain the same genetic information as in the parent, isn't it mom? So DNA replication involves 4 steps. First is origin of replication. Second one is unwinding DNA molecule. Third one is formation of RNA primer. Okay, then synthesis of new strand. So DNA replication involves, first step is origin of replication. Origin of replication. Okay, so replication is the process of producing the new copies of its own or exact copies. Okay, ma'am. So first step in DNA replication is origin of replication. The specific points on the DNA. This is the structure of DNA. The specific points on the DNA where the replication begins. That is the site of replication or site of origin of replication. So in this DNA, where the place where the DNA the replication begins, that is the site of origin of replication. Okay, so during replication, this is one of the strand, this is another strand. During replication, the two strands open and separate at this point, forming a replication fork. This is replication fork. Okay, you all know that how the fork is, isn't it? Now this is replication fork. Okay, so during replication, the two DNA strands open and separate at this point, forming a replication fork like this. This is replication fork. So first is origin of replication. So what is the second step? Unwinding. These two strands are winded each other, isn't it? So during replication, the second process or second step takes place is unwinding of DNA molecule. The enzyme helicase. The enzyme helicase separates the DNA strand. Okay, the enzyme helicase binds the replication site, and this is the replication site or origin of replication site. This here the enzyme helicase binds and it separates the two strands of DNA. So which enzyme is responsible for the separation of two strands? The enzyme helicase. Okay, the enzyme helicase separates the two strands of the DNA. Okay, so where it will take place? This enzyme binds the replication site or origin of replication. There it separates the two strands of DNA. Next, the enzyme topoisomerase separates the double helix above the replication fork. This is topoisomerase enzyme. Here there is the fork, isn't it? Replication fork. This topoisomerase enzyme separates the two strands above the replication. This is below the replication fork, here above the replication fork. Okay, so the enzyme topoisomerase, what is it, ma? Topoisomerase separates the two strands or the double helical structure of DNA above the replication fork. And now the separated DNA strand acts as a template. Okay, this separated DNA the strand acts as a template. Now next step is formation of RNA primer. What is that? Formation of RNA primer. This is the third step involved in DNA replication. RNA primer is a short segment of RNA nucleotides. You know what are nucleotides, isn't it? So RNA primer is a short segment of RNA nucleotides. The primer is synthesized by this DNA template. Okay, this what is called RNA primer is a short segment of RNA nucleotides. The primer is synthesized by the DNA template. Okay, which is close to the replication site. Now the fourth stage, the synthesis of new DNA strand from the parental strand. Okay, what is the first one? Origin of replication. Second one is unwinding of DNA molecule. Which enzyme is responsible for the separation of this two strand? The enzyme helicase. Which enzyme is responsible for the separation of two strands above the replication fork? This is replication fork. Above the replication fork, which enzyme separates the two strands? Topoisomerase. What is called RNA primer? It's a short segment of RNA nucleotide. How it is synthesized? The primer is synthesized by the DNA template. What is DNA template? The separated strands. The separated strands acts as a DNA template. Okay. Then now the next step is the synthesis of new DNA molecule or a new DNA strand from the parental strand. Okay. After the formation of RNA primer, what is called RNA primer? Is a short segment of RNA nucleotide. Okay, RNA nucleotides. After the formation of RNA primer, nucleotides are added with the help of the enzyme DNA polymerase. Okay, after the formation of RNA primer, this new segments are added. Okay, new nucleotides are added with the help of an enzyme DNA polymerase. Okay, ma'am. So new nucleotides are added with the help of the enzyme DNA polymerase, and new complementary DNA is formed from the each parental strand. Now, when nucleotides are added, new strands are formed from the parental strand. Now the synthesis is unidirectional. Okay, so in one of the daughter strand, the DNA is synthesized continuously, and it is called leading strand. Okay, here this strand, the DNA is synthesized continuously, so it is called leading strand. In another strand, what happened is the DNA synthesized in small fragments. These fragments are called Ogasaki fragments. Okay, ma'am. So in one of the strands, DNA formed, this nucleotides formed continuously. This is called a leading strand. In another strand, this nucleotides, in another strand, the daughter strand synthesized as another daughter strand. The small fragments of DNA are synthesized as a small fragments. That is called Ogasaki fragments. This strand is also called lagging strand. Okay, lagging strand. Listen. So in one of the strand, the daughter strand is synthesized as a continuous strand. That is called leading strand. In another strand, the DNA formed or synthesized as small fragments. These fragments are called Ogasaki fragments, and this strand is also called lagging strand. The continuous strand is leading strand. Okay, this DNA synthesized as small fragments. That strand is called lagging strand. Okay, so here this Ogasaki fragments, these fragments are ligated or joined by an enzyme called DNA ligase. Okay, so the fragments are linked together with the help of an enzyme called DNA ligase, or the Ogasaki fragments are ligated or joined by an enzyme called DNA ligase. Now replication, this replication of DNA stops after ligation or after joining these fragments. Replication stops when the fork of the two sides meet at a site called this terminus. Okay, these fork meet at a site called terminus. In this time, these replication stops. Okay, so replication stops when the replication fork of two sides meet at a particular point that is called terminus. Okay, this is about replication. Okay, ma. So replication takes place in 4 steps, or replication involves 4 steps. First is origin of replication. Second one is unwinding of DNA molecule. Third one is formation of RNA primer, and the last one is the synthesis of DNA strand or new strand from the parental strand. Okay, so which here you come to know the name of the enzyme which is responsible for the separation of DNA strand, which enzyme is responsible for ligation. Okay, so which enzyme is responsible for the separation of DNA strand? The enzyme helicase. Okay, mom. So which enzyme is responsible for the separation of DNA strand above the replication fork? Above the replication fork, which enzyme? Topoisomerase. This topoisomerase enzyme is responsible for the separation of DNA strand above the replication fork. What is called RNA primer is a short segment of RNA nucleotides, okay, which is synthesized by the DNA template. Okay, mom. So which enzyme is responsible for the you all know that this nucleotides are added to the new DNA molecule, isn't it, ma? So which enzyme is responsible? After the formation of RNA primer, nucleotides are added, isn't it? With the help of an enzyme. Which enzyme? DNA polymerase. Okay, DNA polymerase enzyme is responsible for the addition of new addition of new DNA or nucleotides. Okay, so new nucleotides are added with the help of an enzyme DNA polymerase. So one of the daughter strand is the leading strand, another is the lagging strand. Why it is leading strand? Here nucleotides are continuously added and it forms a continuous strand. So it is called leading strand. In one of the strand or in another strand, what will happen? The nucleotides are added, or these nucleotides synthesized as a discontinuous strand. Okay, nucleotides synthesized as a discontinuous strand. So it is called, or fragments, like fragments. So it is called Okazaki fragments. All these fragments are ligated or linked by an enzyme called what enzyme? DNA ligase, or it is called ligation. Okay, linked or ligated by DNA ligase. So these are the enzymes responsible for for the replication of DNA. What are the enzymes? Helicase, topoisomerase, DNA polymerase, and DNA ligase. These are the enzymes responsible for the replication. What is called replication? The formation of daughter DNA from the parental one. Okay. Now the significance of DNA replication. This DNA molecule is responsible for, or it helps for the transmission of characters from one generation to the other generation. Okay. What about the significance of DNA, mom? DNA the DNA is responsible for the transmission of hereditary characters from one generation to the next generation. Second one is it contains the information, isn't it? DNA contains the information in the form of nucleotides. These are responsible for the formation of proteins. Next is it controls the developmental process and life activities of an organism. Okay, yes, it is responsible for certain developmental process and also the life activities of the cell. These are the significance of DNA. What about the first one? It carries the genetic information, or it transmits, or it is responsible for the transmission of characters or genetic characters from one, or hereditary characters from one generation to the next generation. Okay, then it is responsible for the formation. It contains, this DNA contains the information for the development of proteins or the formation of proteins. Then the third significance or third importance is that it controls developmental process and the life activities of an organism. Okay, children, you must learn about DNA replication as well as the significance of DNA. Okay, then you must know what is replication, what are the enzymes involved in replication, what are called Ogasaki fragments. Okay, mom. Then the significance of DNA. Okay, children, now you all turn page 271. You all turn to page 271. See, question number this Roman number 3, question number 6. Mark it now. New nucleotides are added. A new complementary strand of DNA is formed with the help of an enzyme, DNA polymerase. That is true, isn't it? Write down true. Okay, mom. Now turn next page 272. Page 272. Fifth question. Roman number 5. Fifth one, name the bond which binds the nucleotides in a DNA. Phosphodiester bond. Okay, phosphodiester bond. Write down. Phosphodiester bond. Phosphodiester bond. Next question, what are Ogasaki fragments? Roman number 6, what are Ogasaki fragments? Answer is there in page 268. Page 268, what are called Ogasaki fragments? Second part, fourth line. So under the heading, synthesis of new complementary strand from the parental strand. Below the heading, second paragraph, third line, in the other strand. It is given, isn't it? You have to strike out that word and write during DNA replication, short segments of DNA are synthesized. During DNA replication, short segments of DNA are synthesized. Next line, the short segments of DNA are called Ogasaki fragments. Okay. Answer is during DNA replication, short segments of DNA are synthesized. The next line, the short segments of DNA are called Ogasaki fragments. Okay, ma'am. The next question. So next question. Long answer question. Roman number 7 first one. Roman number 7 first one you marked already about. First one you marked. Second one, how was the DNA structure of DNA organized? That you marked already. What is the biological significance of DNA? What is the biological significance of DNA? So that is there in page 268, significance of DNA. 3 points are there, isn't it? It is responsible for the transmission of hereditary information from one generation to next generation. It contains information required for the formation of proteins. Third, it controls the developmental process and life activities of an organism. Okay, ma'am? So this next question. Next question, value-based question, Roman number 9, first one, under which conditions does the law of independent assortment hold good and why? This answer you write down, during meiosis cell division, okay, during meiosis, chromosomes assort, during meiosis, chromosomes assort randomly into gametes such that the segregation of alleles of one genus independent of alleles of another gene This is stated in Mendel's third law and is known as Mendel's third law and is known as law of independent assortment. Known as law of independent assortment. Okay, children, then you have to turn page 215 also. There also you have two more questions. 215. Take page 215. 215. Give Roman number 7. Give reasons for the following statements. First one, minerals cannot be passively absorbed by the roots. Answer is there in page 203. 203. Uptake of minerals, last paragraph. Second line, all minerals cannot be passively absorbed by the root. The next up to third line. Third line, concentration of minerals in the root. Okay. Then God cells are responsible. Next question, Roman number 7, second one. God cells are responsible for opening and closing of stomata. It is there in page 202. Under the heading transpiration, third line, the opening and closing of stomata is due to the change in the turgidity of the God cell. When water enters into the God cell, they become turgid and the stoma open. When God cells lose water, it becomes flaccid and the stoma closes. Now, next third one, the movement of substances in the phloem can be in any direction. You have to write down the sentence. The movement of substances in the phloem can be in any direction because the food to reach the plant pods like stem, leaves, flower bud, seeds, etc. Okay, because the food to reach the plant pods like stem, leaves, flower buds, seeds, etc. Next, one more question. Roman number 10. Last question, fifth one. Transpiration is a necessary evil in plants. Explain. Transpiration, answer, write down there. Transpiration is a necessary evil in plants because it is inevitable, but potentially harmful. Loss of water, next sentence, loss of water from the plant results in wilting and cause death of a plant. Okay, and cause death of a plant. And one more time I am telling, transpiration is a necessary evil in plants because it is inevitable but potentially harmful. Loss of water from the plant results in wilting and cause death of a plant. Okay. Okay, children, for your homework, you have to write down the long answer, page 272, long answer, second one. How is the structure of DNA organized? What is the biological significance of DNA? Then they explain the process DNA replication. That you have to these two answers, long answer you are learning and writing in the notebook. Okay. DNA explain the process replication, that also a mark it. Page 268, write down the question there. Explain the process DNA replication. Fully you mark it. Okay. Explain the process DNA replication. From DNA replication is one of the basic process after replication site you mark it. Learn this answer as well as DNA structure. Learn and write it in your notebook. Okay. Thank you children. We shall meet again in the next class.